So it's a great pleasure to be here and present on some new developments and thoughts from University of Munich and the Panacea Consortium on the new drug candidates and the way we are thinking to move them forward. Now, being the first speaker is a bit of a challenge, and I had hoped that you might still be a bit drowsy and not recognize my mistakes, but I was told that the Tmax of caffeine is so short that unfortunately you're probably already at it present. So how does this work? It's just F means forward, I guess. Yes. So what I'm going to talk about today is a brief update on the development situation of both BTZ043, a drug that is developed by University of Munich and the German Center of Infection Research, an update on Q203, a drug that's been developed by Coriand, a Korean pharma company, and a few words on our future plans. Now let's start with BTZ. BTZ has a relatively long and interesting history. It was developed by the Hans Knoll Institute in Jena together with Vadim Makarov, and to cut the long story short, after it was taken over by Alere, development was stopped. Last year, finally, there was a cooperation agreement signed between the Hans Knoll Institute and LMU, the University of Munich, acquiring funds from the German Center for Infection Research to get the preclinical development of the drug finalized. And this year, we received the scientific advice from the German regulatory body, and the preclinical toxicity assessment is completed. And importantly, there is further funding for clinical development until phase 2A from the German government, from DZIF, and from the EDCTP. So the plan for next year is to start GMP synthesis and drug manufacturing for the clinical trial materials, and by the end of next year to start the single ascending dose phase 1A study. And in the year after, the multiple ascending dose phase 1B study, and thereafter again to go into patients and do a phase 2A EBA study within Panacea. So a few words on efficacy, since there was maybe a bit of unclarity, some debates between BTZ and its related substance, also from the benzothiazinones, the PBTZ. So within the German Center for Infection Research, we've repeated the efficacy study to find out what dose gave us the maximum effect of that drug, since that hadn't been tested before. And this is a bit of a classical setup. Bulb C mice were infected with a low-dose infection model, incubated for 3 weeks, and then were treated for 4 or 8 weeks before organs were harvested and colony-forming units were assessed. Now the old data, is there a pointer here somewhere? Check. Sorry. Fantastic. Okay, so the old data published from Vadim Makarov and Stuart Cole showed that BTZ043 gave, some reduction in lung CFU counts if used at a dose of 50 milligrams, while the reduction seen with PBTZ used at same or a bit larger doses seemed to have been slightly larger. Now, in our study here, which was done at Borstel in the mouse facility, we've used various doses of BTZ, as you can see, ranging from 50 milligrams per kg to 1,000 milligrams per kg, just to make sure that we didn't miss any dose range that might change things in a significant way. And as you can see here, after 4 weeks of treatment, the 50 milligram dose gave a similar reduction in CFUs, but the higher doses showed a much better efficacy. And looking at after 6 weeks of treatment and after 8 weeks of treatment, you could see that these 3 high doses from 250 to 1,000 milligrams achieved maximum efficacy or near maximum efficacy. And I would say promising feature is that you can see the INH positive control didn't do as well as BTZ. And as you know, BTZ acts on the DPRE1 enzyme, which blocks arginine synthesis, so is active on cell wall. And the promising thing about this is, of course, that the most active cell wall active compound INH to date seems to be less active than BTZ in this mouse model. Here again, that's the time series. And you can see the three highest doses again group relatively nicely together, so that taking forward the dose of 250 milligrams per kg is seen as the dose giving maximum efficacy. And PK data from this experiment is still underway. Now, safety-wise, it looks really very good. All the preclinical toxicology models showed uncritical results. The no observed adverse event levels doses in rats was very high at 170 milligrams per kg, and in mini pigs was in the same range, so that there is no concern for dose-limiting toxicity at low levels at this point, at least from the animal models. The phase 1A study, starting in 2017, will go forward with 6 doses ranging from 150 to 6,000 milligrams to see what PK and safety data that will give us, and obviously to describe the maximum tolerated dose and, if encountered, dose-limiting toxicities to best characterize the drug going forward. As I said, phase 1B will follow in the year after, and phase 2A again a year later. There was one self-experiment by a person who was professionally linked to the development of this drug, who took the drug at 10 milligrams per kg roughly, and as you can see here, which is a bit small, but we can see here there was a maximum concentration of 600 nanograms per mil achieved in plasma, which was about 300 times the MIC. So that's promising moving forward. Now let's switch to Q203, another new chemical entity, as I said, developed by Coriand. Q203's molecular weight is 557 Dalton. The mode of action is inhibition of the bacterial respiratory chain, the cytochrome BC1 complex. But in addition to working on the bacteria, it's also active in the host and inhibits 5-lipooxygenase. So it is expected to inhibit the formation of pro-inflammatory mediators. Published in vivo efficacy data show here in a mouse model, you can see that here that different doses of Q203 were tested. The blue bars show lung CFUs after 14 days of treatment, and the red bars show 28 days of treatment. And you can see that at 10 milligrams per kg, there seemed to have been a little bit of a lag of efficacy, but after 4 weeks, there was a clear killing to be seen in the range of a bit above 3 log, comparing to before treatment. And there was a clear dose response with 10 milligrams giving the near maximum effect and 2 milligrams being active, 0.4 being not extremely active. And the maximum effect seen was in similar ranges as bedaquiline and INH. Now, I mentioned the activity on the host, and that's the interesting finding here is that comparing lung histopathology after 4 weeks of treatment, you could of course see that these were non-necrotic granuloma mice. You could see that untreated mice showed large lesions, as was to be expected. Mice treated with INH showed a reduction in lesion size, of course, but mice treated with Q203 had an even more marked reduction of lesion size, so you wouldn't be entirely sure which areas are actually lesions in this lung. So this is an interesting feature that we're hoping to learn more about in the future. PK-wise, from the mouse, we've seen that the drug candidate has a very long half-life of 89 hours. So we would expect quite some accumulation of the drug, and a steady state estimated to be reached after 14 days with this long half-life at daily dosing. The NOAL in the dog has been determined as 8 milligrams per kg, and in the rodent at 150 milligrams per kg, which will permit meaningful clinical dosing. The phase 1A study, first in humans, is completed. It was a single ascending dose study. There were 8 persons in each dose cohort, 2 receiving placebo and 6 receiving IMP. The doses used ranged from 10 to 800 milligrams in fasting persons, and the 100 milligram cohort was recalled to receive the same dose again with food to make use of an intra-individual comparison. Due to the long half-life, it was seen necessary to have a relatively long inpatient period, 7 days, 2 clinic visits thereafter, and 1 phone call at day 30 to make sure that the patients were doing well. Safety procedures included physical examination, vital signs, so were pretty standard. The population included 18 to 55-year-old volunteers with a BMI between 18 and 32 kilograms per square meter. People were excluded, also relatively standard criteria, I guess, when they were having concomitant infections, drug abuse, were using concomitant medications that would alter the hepatic cytochrome P450 enzyme activity or change the QT interval, or if they were smokers. The study enrolled from August 2015 to February of this year, enrolled its full cohort of 56 subjects. 15 of those were female due to relatively restrictive criteria on childbearing potential female persons. There's a neat abbreviation for that, but I'm afraid I can't really think of that now. The median ages were between 32 to 46. And all subjects completed the study. Fortunately, there were basically no significant safety findings, neither in the clinical examination nor in the laboratory examinations, and there were also no meaningful changes of the ECG. That was not a surprise because the drug had come clean out of the HERG assay, but it's of course always good to see that confirmed. Now we're at the PK meeting. Let's talk about the PK that was observed. You can see here in the upper graph, this is the Cmax compared to the dose. You can see here that with increasing the dose, there was a not exactly linear increase of Cmax, but the area under the curve did seem to increase in a near linear fashion with increasing dose. Now, surprisingly, there was a very strong food effect seen. As you remember, the 100 milligrams cohort was first dosed in a fasting state and then called back to be dosed again with food. And you can see here the concentration time curve in the fasting state and the one with food, which was tremendously higher. So, in numbers, the Cmax increased by 3.9-fold, and the AUC to the last sampling time increased by 5.13-fold when the drug was given with high-calorie, high-fat meal. I mean, it's a lipophilic compound, so it's probably not such a surprise, but the extent was quite impressive. Now, if we compare the human and the single-dose mouse PK, of course, there's only single-dose human PK to this point, you can see that at least comparing the human 800 milligram fasting and the 100 milligram fat dose, the Cmax was more or less in the same range with the 100 milligram fat dose, achieving a higher Cmax. The AUC were relatively similar, and the half-life was longer than in the mouse in both instances, but longer, of course, in the fat cohort. And to compare this to the MIC90 in most strains, which is 0.2 nanograms per mil, you can see that the drug achieves concentrations that are way above the MIC, which is a good news. But to compare it again with the mouse data that's available, you remember the 10 milligrams per kg was the dose that gave near maximum efficacy. This in the mouse gave about double the Cmax, but importantly it gave a much higher AUC than about threefold higher AUC than the human doses that were tested. Now the 2 milligrams, which also showed quite some efficacy in the mouse, seem to show a similar AUC as shown here. I mean, the AUC of the single dose of 2 milligrams per kg wasn't available, so we'd have to use the steady state AUC up to 24 hours in the dosing interval, which is similar as the AUC of 0 to infinity after a single dose. So to point that out again, these are always the kind of animations that come up when you've already read the slide. But yeah, as you can see, the conclusion is that with the current human dosing, the exposures achieved are in an effective range as compared to the BALB/C mouse. And we would, of course, want to see higher exposures in the human if toxicity permits. As I said, so Q203 was tolerated and safe at all doses tested. There was no dose-related or dose-limiting toxicity encountered. And the human PK achieved levels which were effective in the mouse, and the strong food effect was mentioned. Currently, Coriand is conducting a phase 1b multiple ascending dose study with the doses of 20, 50, and 100 milligrams, which are given daily for 14 days with food. And I'm happy to say that the first dose cohort has already been completed. That will be followed by a phase 2a study, which will, as far as possible, use the 14 plus 14 SMART study design that I'm going to talk about in a second. And that's what I would like to switch to, if I still have a couple of minutes left. The main preclinical development questions that are open and unanswered at this time, to me, seem to be the role of persisting organisms in drug development. We somehow seem to feel that they are important, but what is the evidence that is out there? The role of necrotic granuloma mice in preclinical drug development, and I think Deborah and her team are doing very important work on this to validate this more human-like mouse model for clinical development. And finally, we have a lot of interesting information on drug lesion concentrations, which at least I don't really know yet how to factor that into preclinical drug development. First, a few words on the persisting non-cultiviable bacteria. And I like to quote here a paper by Yan-Min Hu and Professor Coates from St. George's in London, who did a mouse study on high doses of rifampicin and compared here a 10 milligram and a 50 milligram dose level given for 8 and 14 weeks. And they compared the plate counts of classical plating CFUs on lung after this treatment period to broth counts using resuscitation promoting factors, so culture filtrate from log phase cultures that would be expected to wake up the persister phenotype bacteria in the organs which are not growing on this solid media plate. And as you can see, the plate counts here were consistently lower than the broth counts using RPFs, at least here in this lower dose cohort treated for 8 and 14 weeks. And interestingly, after 14 weeks of treatment, the organs were sterile when plated on solid agar plates, but when using liquid media with RPFs, there was still quite some bacterial growth to be detected. And at the higher doses of RIF, sterility was achieved in both culture methods. And you will probably not be surprised that after steroid treatment in their modified corneal mouse model, most of the mice in this upper cohort showed a relapse, while none of the mice in this lower cohort with 50 milligrams per kg. So you can see, to me, this demonstrates that sterility, as is already known, sterility in solid media plates does not predict sterilization of the lesions and prevention of relapse when there is still detectable growth using RPFs. When you look at human data, that gives information on these persisting phenotypes. There is one important or interesting study published just recently from Cape Town, where they looked at mRNA positivity at end of treatment and found that around a third of the cured patients still had detectable mRNA in their sputum at end of treatment, and also, well, similar numbers, similar percentages of patients who had recurrent disease. So in patients, this data is less clear, it seems, than in mice. But what we've also seen is that Derek Sloan and Jerry Davies have published from Malawi a cohort of patients who had a poor outcome if they had a higher positive percentage of lipid body positive cells in their sputum at week 3 to 4. So this, of course, is not a direct association to a persistent phenotype, but is some indication that this cell type may play a role. Now again, at least the animal data are straightforward here, and the human data might be a bit confusing. It seems next year I have to finally switch to a different kind of joke. But conclusions to be drawn from that, and these are only more or less hypotheses on some coming from some pieces of available evidence. Conclusions we would draw from that is that we would probably shift to the necrotic granuloma mouse model at this point, without very good evidence, and we're still waiting for the CPTR validation work on that. We would probably want to know more about the bacterial burden in those mice, comparing solid agar growth phenotype versus the total live bacilli, and we would, of course, very much like to use molecular markers to do that. We would want to establish a PK/PD relationship using individual mice already, and sampling and analysis of that has been established and is working. And we would want to have information on lesion drug concentrations and watch the field where that goes. Now, a few words to finish with our early clinical study design. And I'd just like to list here the early clinical studies that have been done on bedaquiline, as I've found them from clinicaltrials.gov. You can see that there was really a number of studies done on bedaquiline, a very thorough and admirable work to characterize that drug before it went into, or along with it going into the later phase clinical trials. And we would, of course, hope that this really high number of studies could somehow be abbreviated and shortened. And as you probably know, we are very fortunate at the Panacea Consortium to have received renewed funding and would like to put that into reality. So looking at the objectives of these early clinical studies, and let's call this a classical EBA study design for the moment. Such studies have the objective of establishing the activity of a new drug in monotherapy, looking at the PK/PD in the target population, and of course safety and ECG in this target population as well. And the TB Alliance have made a very important step of doing these studies not only as a monotherapy, but also in combination with other anti-TB drugs to gain additional insight into activity and PK/PD of their promising new combinations. We would hope that these studies would then also give us information on drug-drug interactions with other TB drugs, so that we would be able to design our phase 2b studies in a more informed way, and also drug-drug interactions with potential concomitant medications, most important being, of course, HIV drugs in our patient population. Now, the classical phase 2a study design is depicted here patients would be characterized in a pre-treatment period and then randomized to either receive the classical standard of care control treatment or to different doses of the investigational drug, mostly as monotherapy. And over this 14-day treatment period, safety and efficacy in terms of the drop in bacterial counts in the sputum would be observed. Now, we would like to add a second randomization step, and that's where the abbreviation SMART comes from, sequential multi-arm, multi-randomization trial. Hope I got this right. Patients who've been receiving the IMP for the first 14 days would have a second randomization done and then go for another 14 days of combination therapy, of course it being completely unethical to dose somebody for 28 days as monotherapy. So they would be randomized to either receive the standard, we say, backbone of isoniazid, RIF, and pyrazinamide, or a novel backbone that is to be determined based on what the scientific field discovers and describes. And this will, of course, give us some hint of efficacy if we are able to compare the decline in bacterial count between day 1 and day 14 to the decline in bacterial count in the later time period. But importantly, it will give us information on pharmacokinetics. As you can see, that we would do a pharmacokinetic assessment at the first dose, at day 14 at the steady state, so we would be able to compare accumulation here. But then when we dose it together with a novel backbone or with HRZ, we would be able to see how the steady state concentrations are altered if the drug is given with other medicines concomitantly, other TB drugs. Now the next question is about other concomitant medications, including HIV drugs, and the problem here is, of course, that we cannot all include them and dose patients with all these drugs that would confuse our outcomes. So we thought we might use a method of giving the patients a probe drug cocktail of underdosed drugs with a very clear metabolism, like midazolam, which is metabolized by CYP3A4, or caffeine, which was already mentioned today. So this cocktail is still to be composed, but the main objective is that there should be drugs with a clear liver enzyme they are metabolized by. So we would give that to the patients before they receive any IMP, so to say, to characterize their metabolic phenotype before they receive drug. We would give that to them again at steady state so that the blood sampling for PK can be done together with the PK sampling of the IMP. There we would see how the metabolic profile is altered by the IMP alone. And then here we would do it again and see how it is altered to see when the patient is dosed with companion drugs. So that is basically what we are planning to put into practice in the next 2 months. And yeah, I think next year we will probably know more, at least whether it's possible to put all this into one study protocol. If not, whether we'll have had the chance to get the patients through on it. With that, I'd like to thank a number of people and institutions, University of Munich, Michael Hölscher and Julia Dreisbach, who are leading the development of BTZ, funded by the DECIF, our colleagues at Coriand and at the Research Center of Borstel, and finally the EDCTP, who've been deciding to funding us again. Thanks for your attention.